All right, welcome to Chem 313 Lecture 4. So today's topic is going to be protein structure. And when I use the word structure, what I'm really talking about is conformations. We are not talking about covalent structure, although that is also important. What we're talking about is the three-dimensional shape. So protein structure is a fairly complicated topic, and if you want better practice problems for this subject, I'm going to suggest that you have a look at homework 2 and worksheet 3. Both of these have some practice problems that are related to the topic of protein structure. And I'm also going to put out a short additional lecture, I call it lecture 4.5, which is a brief demonstration of how 3D structures of proteins are viewed computationally. And it's a handy resource if you want to get some additional practice. So to review, last time we talked about amino acids, which are the building blocks for proteins and peptides. And the term protein or peptide, they can be connected by the generic term polypeptides. And we said amino acids are molecules that have an amine group, a carbon that's referred to as the alpha carbon, a carboxylic acid group, and a variable group we call the side chain. And we note that the amine has a positive charge and the carboxylate has a negative charge at neutral pH. And we said that you can take multiple amino acids and polymerize them to give us polypeptides, which are molecules that look like this. What we have are these amide or peptide linkages connecting each of our amino acids. And we have side chains hanging off the side. This slide is just a review of the last slide we showed at the end of the previous lecture. We said that peptides are short polypeptides with a few amino acids in them. And proteins is the term that is reserved for the longer polypeptides. And we use this molecule shown here as an example of what a polypeptide molecule looks like. You'll notice that we have what's called a main chain, which contains the alpha carbons, the amines, and the carboxylate carbons. And then our R groups, which we call the side chains, hang off the side of the main chain. We said that every polypeptide is a repeating sequence of peptide bonds and a repeating sequence of side chains as well. And we said that each polypeptide has an end with a free amine, which we call the N-terminus. and an end with the free carboxylic acid, which we call the C terminus. And we refer to the sequence of amino acids as a primary structure. So it's important to understand a few things about our polypeptide chains. You can think of a peptide or a protein as a chain of peptide bonds, right? Because we see peptide bonds linking every single amino acid in the chain. So when you look at a peptide bond, what kind of intermolecular forces do you think are going to be active with this group? Remember, we are always going to be in water in biology. Every molecule that we see, with only a few exceptions, is going to be completely surrounded by water at all times. So most likely what's going to happen is our peptide bonds are going to tend to form hydrogen bonds. All right, what they form hydrogen bonds with, we're going to talk about momentarily. But regardless of the identity of our side chains, the peptide bonds are always the same, and the peptide bonds are always going to form hydrogen bonds. But there's a different way you can think of peptides and proteins. You can also think of them as a chain of different side chains. And side chains are a little bit different because the intermolecular forces that each of those can take part in vary. It depends on the type of side chain that you have. So for example, lysine and arginine, because they're positively charged, they can undergo ion-dipole or ion-ion interactions with other charged molecules. But then nonpolar side chains like, say, leucine or alanine, those can only take part in hydrophobic interactions. And so this creates a bit of a problem. The peptide bonds and the side chains may have different intermolecular force capabilities. And so which of these is going to win out? And the whole theme of today's lecture is the interplay between the types of intermolecular forces that the peptide bonds want to take part in and the types of intermolecular forces that the side chains want to take part in. These factors determine the conformation of our protein. All right, so we're ready to talk about full-size proteins. Right, so a protein, the proper definition of a protein, is a long polymer of amino acids that fold back on themselves in a well-defined three-dimensional shape. So why do they do this? Well, if you imagine just a short peptide, what would a short peptide look like? Well, I'm going to come up with an arbitrary short peptide. I'm going to use one-letter amino acid codes. Let's say that our short peptide is L, A, E, F, G, A, K, L, D. So what we have here is what? A 9 amino acid peptide. And I refer you to the amino acid table for the one-letter amino acid codes. And once again, you're not going to be required to memorize those, but if you could at least remember which group each amino acid falls into, you're going to do a lot better in this course. All right, so for any peptide, we have an N-terminus, which is going to have a positive charge at neutral pH. We have a C-terminus, which is going to have a negative charge at neutral pH. And then what do we have? Well, we've got a bunch of side chains. All right, so what kind of properties do these side chains have? Leucine is nonpolar. Alanine is nonpolar. E-glutamic acid is negative at neutral pH. Phenylalanine is nonpolar. Glycine is nonpolar. Nonpolar. Lysine, positive. Nonpolar, negative. All right, so what do we observe in this peptide here? And by the way, I've drawn the orientation of the side chains arbitrarily. They don't literally alternate up and down like this, except in certain specific cases we'll see later. What do we notice? We have nonpolar side chains here that are going to be unable to hydrogen bond with water. Water is our solvent, and therefore they're going to tend to form droplets or shield themselves from water. How does that happen? We'll see later on. But then down here we have charged groups, and these charged groups will interact with water by the ion-dipole effect, ion-dipole interactions, or with each other. And then in between each of these amino acids, we have peptide bonds which must form hydrogen bonds. So the question now becomes how to satisfy all of these intermolecular force needs. What ends up happening is that the molecule folds up into a compact three-dimensional shape. So what we're seeing here is myoglobin. Myoglobin is a protein we're going to talk about more next week. We're showing two different views of the same molecule. One on the left is what's called a ribbon diagram. One on the right is a space-filling diagram. So in the ribbon diagram, we have some side chains that are shown in blue as balls and sticks. The ribbon is the main chain, or the backbone. And we're going to see examples of other protein structures in ribbon diagrams later on. So the reason that these compact three-dimensional structures form is that this is the way to maximize the satisfaction, if you will, of all of our different groups in terms of their free energy. In other words, it's a minimum of free energy. Everything forms the best possible intermolecular forces that it can when the molecule is folded up into this structure. Now, what we're going to do now is take a look at how these structures actually form. All right, let's imagine that we have a polypeptide chain. And in our polypeptide chain, we're going to have peptide bonds. We're going to have alpha carbons with side chains on them. And then we have another peptide bond afterward, and the chain keeps going. Now, notice that here we have one peptide bond on either side of the alpha carbon. Now, an important thing to notice about peptide bonds is that peptide bonds are planar. They're planar, and they're usually in the trans conformation. Why is that? Well, the reason for that is that if you look at a peptide bond, it's possible that we can draw a resonance structure where the lone pair electrons from the nitrogen get pushed down into the carbon-nitrogen bond, and those on the pi bond pop up onto the oxygen, which gives us a structure that looks something like this. And in this resonance structure, we have sp3 hybridization on the carbon and the nitrogen, and therefore we have a planar geometry. Now, in reality, this resonance structure is a minor contributor. The molecule looks more like this. Nonetheless, because of resonance, we have planar peptide bonds. And this central bond cannot rotate, not easily. So when we said last time that you can think of a polypeptide chain as a repeating string of single bonds in CC, in CC, in CC, it's not as if it's truly a string of single bonds. The nitrogen to carbon bonds have some double bond character to them. So really only 2 out of 3 bonds can freely rotate. But still, that's 2 out of 3 bonds, right? So we have a chain that is flexible. And we do have free rotation around these bonds here, the 2 bonds that connect to the alpha carbon. So what does this mean? This means that the C alpha to nitrogen and C alpha to carbon bonds will rotate to change the conformation of the backbone. So what that means is that if you have an extended polypeptide chain that looks something like this, it's not really going to look like this normally. What it's going to do is it's going to fold up into some sort of coiled conformation that looks more like what's down at the bottom. And what we've done in that picture is just color-coded every amino acid according to its properties. So what ends up happening is, once again, the C alpha to N and C alpha to C bonds rotate to provide a well-defined conformation of the chain. Now, what is the conformation that it's going to adopt? So I'm going to tell you right now that each protein has pretty much one and only one conformation that it strongly, strongly favors. There may be slight variations within that conformation, but for the most part, most proteins form one structure. So what decides which structure it's going to form? Well, it's an optimization problem. The best conformation is the one that has the least steric hindrance, it has the most peptide bond hydrogen bonding, and that optimizes side chain intermolecular forces. All right, so last time we said that there's something called primary structure, which is the sequence of amino acids. This defines the chemical structure of our polypeptide chain. There are different levels of protein structure. There's secondary structure, tertiary structure, and quaternary structures. Each of these defined the conformation of the polypeptide chain in different senses. So we've already talked about what primary structure is. Let's talk about what secondary, tertiary, and quaternary structures mean. Secondary structure, we can abbreviate it as 2 with a degree sign. Secondary structure is the manner in which a protein chain folds up locally so that the CO and NH bonds of the peptide bond can form hydrogen bonds. Secondary structure, in other words, refers to the pattern of hydrogen bonding between peptide bonds in the protein chain. So we said on the previous slide that the optimum structure is the one that maximizes peptide hydrogen bonding. Well, secondary structure is the different ways in which the peptide chain can do this. So there are essentially 4 common types of hydrogen bonding patterns that we're going to see. There are more, but there's 4 that I expect you to know. These are called the alpha helix, the beta sheet, the turn, and the coil, which is also called a loop. And we're going to look at each one of these in that sequence. All right, let's look at the alpha helix first. The alpha helix is what you get when the backbone folds up helically and the hydrogen bonding is between amino acids 4 positions apart. The way an alpha helix is drawn in a ribbon diagram is like so. I have a hard time drawing these efficiently. Something like this. So once again, the ribbon represents the backbone or the main chain. The flatness of the ribbon is supposed to represent the flatness of the peptide bond, because remember the peptide bond is planar and therefore it tends to be rather flat. So the peptide bond lies in the plane of the ribbon. All right, so if your peptide bond lies more or less in the plane of the ribbon, what you end up with is a molecule that looks like this if you were to zoom in on each of the individual atoms. What we have are peptide bonds. So here is our NHCO. And notice that each NH is lined up with a hydrogen bond acceptor 4 amino acids away in the sequence of the protein. And likewise, each carbonyl is lined up with a hydrogen bond donor 4 amino acids away in the sequence. So in an alpha helix, we have hydrogen bonds that are aligned along the length of the helix between successive turns. They're parallel to the axis of the helix, in other words. Okay, your textbook and all textbooks make a huge deal out of the dimensions of the helix, which I don't personally care about. If you really, really care, we have about 3.6 amino acids for every turn. Or you can think of it perhaps better as we have 7 amino acids for every 2 turns. So what that means is that a 2-turn alpha helix like I showed here would be roughly 14 amino acids in length. Okay, so we said that our hydrogen bonds lie along the length of the helix between successive turns when our polypeptide coils up into an alpha helix. But where are the side chains? Well, the side chains stick out. So if you were to look down the helix, that's what this picture is supposed to be showing, you would notice that all of your groups form this big repeating cycle along the exterior of the helix. So our R groups are going to be pointing outward. And that means that everything that's on the inside is essentially shielded from the water, because remember, water is everywhere. We've got water all around the outside, unless we have this structure that's buried within the interior of a protein globule. What this figure down here is supposed to show is the hydrogen bonding pattern. So what we have is a hydrogen bond between the carbonyl of amino acid 1 and the amine of amino acid 5. And the same thing would be true for the next carbonyl. This would be hydrogen bonded to the NH of amino acid 6. This is just a different diagram of the ribbon of an alpha helix. I think it's a somewhat clearer picture. But notice what happens here. When you have 2 separate amino acids that are on adjacent turns, notice that the R groups are relatively close together. This would be a top view of the same thing. So in other words, amino acid 1 is not directly above, but it's fairly close to amino acid 4, right? And so what that means is that R1 and R4 oftentimes can interact with one another. So this is something you don't always see this in alpha helices, but you do see it frequently enough that it's worth mentioning. And that is amino acids that are 3 positions apart may have side chain interactions. However, the absolute position of the R group repeats every 7 amino acids, right? Because of our rule that we said that you can think of an alpha helix as having 7 amino acids per 2 turns. So that means that amino acid 1 and 8 are almost directly on top of one another. However, the R groups are not close enough together to interact because one of them is all the way up here and the other one is way down there. All right, so which types of proteins form alpha helices? So I'm going to give you a general rule here. This is never a hard and fast thing. It's a general rule of thumb. Because our side chains are splayed out along the exterior of the helix, alpha helices are preferred by amino acids with large side chains. Now, that does not mean that you will never ever see an alanine in an alpha helix. You can and you do. What we're saying is that all else being equal, a stretch of polypeptide with a lot of large side chain amino acids like arginine and tryptophan, that's more likely to form a helix than anything else. There's 2 amino acids that have special effects on alpha helices. And those 2 amino acids are glycine and proline. Both of these are oftentimes said to be so-called helix breakers. What does that mean? Well, what that means is that if you have a stretch of alpha helix like this, if you have glycine, remember on glycine, that's the smallest amino acid. The side chain is just a hydrogen atom. Because of that, glycine is flexible. And so glycine tends to terminate a helix. Proline does the same thing, but for a different reason. So proline essentially introduces a kink. And that's because of the shape of the proline side chain. So remember, proline is the one where the side chain is actually connected to the main chain in 2 places. So if you have proline after a stretch of alpha helix, typically what it's going to do is, once again, either terminate the helix or it's going to introduce a rigid bend that will make the helix go off at a funny angle like that. Okay, next type of secondary structure, the beta sheet. So beta sheets come in 2 different flavors. These are extended segments of polypeptide chain lying side by side. The ribbon diagram for a beta sheet looks like this. We draw them as flat arrows. And then the arrow always points from the N terminus to the C terminus. So we have segments of polypeptide chain that lie side by side like so. And then obviously, since it's all one molecule, this has to somehow coil back around to come over the other side, which is fine. And then our hydrogen bonds are in between the strands like this. So our hydrogen bonds are perpendicular to the backbone. So I said that these come in 2 different flavors. The one that I just drew here is what's called a parallel beta sheet. And in parallel beta sheets, all segments point the same way. So what do I mean by all segments point the same way? I mean that the N terminus and the C terminus on each strand lines up. This is a ribbon diagram down here for a parallel beta sheet. In an anti-parallel beta sheet, the segments alternate. So we have the N terminus and the C terminus, but the next one down, we have C, N, and then NC. You can also have mixed beta sheets, which would be something like this. Those are less common, but they do exist. All right, so terminology time. Each individual segment of chain that's part of the beta sheet is referred to as a beta strand. The whole thing is the beta sheet. So the beta sheet refers to the entire collection of segments that are hydrogen bonded together. Each one extended segment is a beta strand. So regardless of whether you have a parallel or an anti-parallel beta sheet, notice where the side chains are. The side chains alternate up and down above and below the plane of the sheet. So if our sheet is more or less flat, what we have is an R group that points down, and the next one it points up, the next one down, the next one up. And if you were to look at this from a side view, this is intended to be looking at the thing kind of edge on, we would notice that our R groups appear in rows. And then the same thing on the bottom. All right, so in beta sheets, our side chains are packed together in rows. And that should give you a clue as to the types of amino acids that tend to prefer beta sheets. So just like with alpha helices, we can come up with rules of thumb, but there are no hard and fast rules. You will find almost any amino acid in a beta sheet if you look at enough protein structures. However, there are some amino acids that prefer beta sheets. So beta sheets tend to be preferred by so-called branched chain amino acids. That would be valine, isoleucine, and threonine. And it's also preferred by asparagine and glutamine. And the reason that asparagine and glutamine like beta sheets is that these ones can hydrogen bond side by side if they're next to each other in rows. So oftentimes you will see the side chains as well as the backbone peptide bonds hydrogen bonding with one another in a beta sheet as well. Or occasionally you'll see ion pair interactions between side chains. One amino acid that you will never find in a beta sheet is proline. That's nearly a hard and fast rule. And the reason for that is, once again, because proline is the only amino acid where the side chain is bonded to the backbone at two places. It's just too conformationally restricted to form a beta sheet. All right, our next form of secondary structure is what's called a turn. So a turn is pretty self-explanatory. It's a stretch of about 4 amino acids, all right, but it can be anywhere between 3 and 6, or even rarely 7. And essentially what happens is the chain does a 180-degree turnaround. In most turns, there is a hydrogen bond between the peptide bonds of the first and the last amino acid in the turn. So that's what this diagram is supposed to show you over here. We have C alpha of amino acid 1, its CO group from its peptide bond. Here is C alpha of amino acid 2 with the R group pointing up, amino acid 3 with the R group pointing up, and then finally amino acid 4 whose NH group hydrogen bonds with the first group. So what we have is something that looks kind of like this, with a hydrogen bond sort of capping it in place. And it does a more or less 180-degree turn. So turns are oftentimes seen between two strands of an anti-parallel beta sheet. And as an example of that, I show you this protein structure right here. What we have is a zoomed in version of the turn that's right at this corner between these two strands of this beta sheet. So this protein is what's called a beta barrel protein. It has an anti-parallel beta sheet that sort of folds back into a big tube. And look at what we have down here between these two strands. We have 4 amino acids that form this nice little turn all together. And this picture shows you a zoomed in view of what the side. I don't have a picture for it, but another place that you'll sometimes see turns is between 2 alpha helices. Okay, we said in our earlier slides that proline and glycine are not very common in alpha helices, and we said that proline is essentially never seen in a beta sheet. Well, in turns, proline and glycine love them. Proline and glycine are overrepresented in turns. Why is that? Well, that's because proline is essentially already bent. It's halfway there. And glycine, because it's highly flexible, it can accommodate almost any structure that you want. So turns don't always have a proline or a glycine, but as often as not, they have at least one of those. And if you ever see a proline and a glycine together, bet you $50 that's a turn. So you can see in this structure down here at the bottom, we have a glycine in position 2. Because glycine has a very small side chain, so it's going to be flexible and it can accommodate this rigid turnaround. Okay, our last secondary structure is kind of the default secondary structure. It's sort of a non-secondary structure, and that's the loop or the coil. So in the loop or the coil, the peptide bonds don't hydrogen bond with each other. Instead, they tend to hydrogen bond either with the surrounding water or occasionally with other molecules, side chains, ions, something else. So more often than not, loops or coils are exposed to water. They also tend to be flexible. And why is that? Well, the reason is because you don't have hydrogen bonds, you don't have peptide bonds hydrogen bonded to other peptide bonds. You really have nothing that's really tying this loop in place. So a place where you tend to see loops is between other secondary structure elements. So for example, in some proteins, you may have a helix and then a turn and then another helix. And then we have a loop, a beta sheet, another loop, another helix, something like that. So these would be our loops. In the middle would be a beta sheet, and over here would be the helices. Another time you'll often see loops is between different strands of a beta sheet. So for example, if we have a parallel beta sheet, as I mentioned earlier, once our chain comes out that beta strand, it has to go somewhere in order to form this other strand. And so what often happens is it'll sort of loop back behind and come out the top like that. So in that case, we have a loop connecting two strands of a beta sheet. So that basically does it for secondary structure, right? So to summarize secondary structure is all about satisfying the peptide bonds. And by satisfying, I mean fulfilling their need for hydrogen bonding. And remember, the optimal secondary structure is the one that maximizes hydrogen bonding between the peptide bonds while minimizing steric clash. And what that optimal secondary structure is going to be really depends on the protein. It depends on your amino acid sequence. It depends on your amino acid content. It depends on a lot of things. Okay, so where we're going to go with this now is into the realm of tertiary structure. So what's tertiary structure? Tertiary structure is an assembly of secondary structure elements that give the protein an overall shape. And the primary driving force for tertiary structure is to keep nonpolar amino acids away from water. Here's what I think you should, how you should keep secondary and tertiary structure clear in your mind. Secondary structure is all about peptide bonds. Tertiary structure is all about side chains. It's all about optimizing side chain intermolecular forces. And by side chain intermolecular forces, I could mean hydrogen bonding with water. I could mean nonpolar side chains being kept out of water, and positive and a negative charged side chains interacting with each other by ion-ion interactions. So this diagram illustrates the distinction between primary, secondary, tertiary, and quaternary structure. Primary structure is your sequence. Secondary structure is how that sequence folds up so that the peptide bonds will hydrogen bond. Tertiary structure is how the different secondary structures pile up to give you a three-dimensional shape. Quaternary structure is very similar, but we're going to talk about that last. There's two classes of tertiary structures, fibrous and globular, and most proteins are globular. But regardless of whether your protein is fibrous or globular, the forces that define the tertiary structures tend to be the same in both, and so I'm going to go over fibrous proteins first just to illustrate how tertiary structure works. All right, what we're seeing here is collagen. Collagen is the best known of all fibrous proteins. And essentially, our polypeptide chain of collagen coils up into this helix-like structure. This is not an alpha helix. What this type of structure is called is a polyproline 2 helix. That's a rare sort of secondary structure that we don't have to know about for the purposes of this class. So notice that this helix is left-handed. Alpha helices are typically right-handed. And this structure is too sort of stretched out the different side chains don't hydrogen bond with one another along the lengths of the helix here. So this is not an alpha helix. Regardless, it is sort of a coiled up structure. And what happens is we have 3 segments. In the case of collagen, it's actually 3 separate molecules that coil together. And you can actually see this under an electron microscope, which is what this picture is trying to show. You have one molecule of collagen that coils up with 2 other to form a bundle, and then those bundles coil up into super bundles, and then the super bundles coil up into big, thick collagen fibers, which you can even see under a standard microscope, not just electron microscope. Under electron microscope, you see this characteristic banding pattern. So why does collagen do this? It's all about the side chains. So if you were to look at the sequence of collagen, it's got this regular repeating sequence: glycine, hydroxyproline, regular proline, alanine, hydroxyproline again, proline again, and then it keeps on repeating like that. Okay, so what are our side chains? Glycine, the side chain is just a hydrogen. Hydroxyproline, it's an aliphatic ring with an OH on it. Proline is just the regular ring. Alanine is a methyl. Hydroxyproline again. Proline again. All right, so we have this repeating pattern. This is how the different chains pack together in this coil. We have our glycine, whose R group, just an H, is pointing to the interior of the structure. Now, does that make sense? I would say it does, because glycine is a nonpolar amino acid, and all along the exterior of this structure, we have water. So because this cannot hydrogen bond with water, because of the hydrophobic effect, it's going to tend to be sequestered. Right? Because water cannot form hydrogen bonds with it, water is going to tend to push it to an interior structure where water doesn't have to form that unfavorable cage-like arrangement around the surface of it. Now, hydroxyproline can hydrogen bond with water, right? It's got a hydrogen bond doning group. So that's why hydroxyproline has the OH facing out. That's supposed to be coming out of the screen at you. Right? So our hydroxyproline does face water. Proline is nonpolar, so it tends to face away from water. Now this one is exposed. I'm going to talk about that in a moment here. Alanine, once again, faces the interior, away from the water. Hydroxyproline again, OH is pointing out. Proline again, right? So what do we see here? We see that nonpolar side chains face inward, and hydrogen bonding side chains face outward. Now, if you're paying attention, you may think, well, wait a minute, what about proline here? Proline is nonpolar, it doesn't form hydrogen bonds, and yet it's sticking out into the water. How can that be? Well, the answer is you are right. That is an unfavorable thing to happen. So what does the system do in order to minimize this problem? What the system does is it takes 3 of our collagen chains and twists them together like here in order to bury the prolines. And therefore, by forming these big, complicated cables, all we're doing is trying to bury nonpolar amino acids away from water and keep the polar hydroxyprolines exposed to water. So globular proteins, this is what most proteins are. Fibrous proteins aren't that common. So globular proteins are spherical, if you don't look at them too carefully. Sometimes they can be a little bit elongated. But, the terms are self-explanatory. Fibrous proteins are fibrous and globular proteins are globular. So this is our friend myoglobin again. All right, so what do you notice about these globular proteins? So the globular proteins don't look like the fibrous proteins, but the principle is exactly the same. What we've done is shown the nonpolar side chains in yellow. And if you look at the ribbon diagram of myoglobin with the nonpolar side chains shown in yellow, what do you notice? Most of the nonpolar side chains are going to be on the inside of the structure. This is a space-filling diagram, and what do you notice? You barely see any yellow at all. Everything that's on the outside is gray, which is either backbone or polar amino acid. Don't worry about what the red is. We're not going to talk about that yet. So the idea of globular proteins is globular proteins are going to fold into a compact shape to keep nonpolar amino acids on the inside away from water. Which is simply what the hydrophobic effect is. So what does this look like in real proteins? Well, let's imagine that we look at the length of an alpha helix. So what we have here is the protein flavodoxin. And what we're doing here is we are looking at the sequence of this alpha helix that's shown in red. So if you were to look down the length of this sequence and graph where each of the R groups is on this molecule, what would you notice? You would notice that in orange are these nonpolar amino acids. Yes, there's one on the other side, alanine. Don't worry about that. So what we have is nonpolar amino acids that are on one side of the helix. And on the other side, we have mostly polar or charged amino acids. So how is this alpha helix going to satisfy the need for these nonpolar R groups to be away from water? What's going to happen is that in the intact protein structure, when it folds up, it's going to lie against the surface of the protein so that all of these nonpolar R's point inward and are away from the water, which is all around the outside. Now, globular proteins have a lot more structural variety than many fibrous proteins do. So you can have small ones, you can have big ones, you can have all kinds of different shapes and sizes. I like this diagram because it shows all of these different proteins on a constant scale. This is drawn to scale. So we have insulin, which is a very small protein. We've got molecules like myoglobin and lysozyme, which are kind of medium sized. And then at the bottom, we've got some bigger proteins. So one thing to notice is that many proteins have more than one molecule in them. So all of these are globular proteins. However, these four at the top have a single polypeptide chain. And here the terminology gets a little confusing. These proteins are said to be monomeric. Now, I know we said in the first lecture that a monomer is the same thing as a building block. Yes, that's true, but in the protein world, we tend to use the term monomeric protein to refer to proteins that have a single polypeptide chain. Now, some proteins have two or more polypeptide chains, in other words, two or more polypeptide molecules stuck together. And those are said to be multimeric proteins. So in this case, your protein, so to speak, is actually two polypeptide molecules, right? We use the definition protein for the functional unit. The whole thing together, since they're always found together, we treat it as a single entity, but it's actually two molecules. Insulin is an example. Insulin is a dimeric protein. It's got two chains. It's got an A chain and a B chain. And A and B are different, so it's said to be a heterodimer. So insulin is one of the smallest multimeric proteins. Another multimeric protein is hemoglobin. Hemoglobin is said to be a heterotetramer. Tetramer meaning it has four chains. Hetero meaning they are not all the same. So hemoglobin has two chains that are called the alpha chain, and it has two chains that are called the beta chain, right? And where the term alpha and beta comes from, who cares? Whoever discovered it got to name those chains whatever they wanted to. They just named them alpha and beta. Immunoglobulins are the same way. These are also heterotetramers. They're just much larger than hemoglobin. Immunoglobulins have two heavy chains and two light chains. Glutamine synthetase is a homododecamer. It has 12 chains and they're all the same. And they're stacked together in this double ring structure that you see on the lower right. So this leads us to the idea of what is a chain. What's a chain? So in the protein world, a chain is a single polypeptide molecule. A protein refers to a single functional unit. One or more chains. So now the question becomes, why would certain molecules stack together like this? Why would some polypeptide chains only be found piled up on top of other polypeptide chains? And the answer is the exact same reason that proteins have tertiary structure: to satisfy the side chains. So proteins like this that have multiple chains are said to have quaternary structure. So quaternary structure is exactly the same thing as tertiary structure. It's all about the side chains interacting with one another. Okay, but it's between different chains. All right, a couple little loose ends that I wanted to tie up before we conclude the lecture. Your textbook talks about these terms that come up frequently enough that I think it's worth addressing in the context of this course. There's this concept called motifs or folds. Motifs and folds, these are sometimes called super-secondary structures. And what these are is small little groups of secondary structures that are oftentimes seen together. The difference between a motif and a fold is subtle. Usually motifs are smaller and folds are bigger, and we're not going to get into the fine points of how you tell a motif or a fold. For the purposes of this class, you can think of them as the same thing. So for example, the beta-alpha beta loop. This is the type of structure where you have a strand of a beta sheet. The chain then folds behind itself to give an alpha helix. and then you have another strand of the data sheet behind it. That's what's called the beta alpha beta loop. So you will see this in several different proteins that have this kind of structure. It's relatively common. So we call that a motif. The beta barrel we already saw, that's also a motif. That's where you have a anti-parallel beta sheet that coils back on itself into a tube, and it's essentially hollow on the inside. Related to motifs or folds are the concept of domains. So a domain you can think of as a small protein within a protein. So it's a little globular subsection of a protein that can fold and act independently. So oftentimes domains are connected to the rest of the protein by a loop. Not always, but oftentimes. So this protein would have two domains. And there's a loop connecting the two. This one at the top would be beta sheet rich, and this one at the bottom would be alpha helix rich. So domains are not always easy to spot in a protein. If I ever give you a question where I'm going to say, how many domains does this protein have? I'm going to make it very, very obvious. But the key feature of a domain is this. If you could cut it out of the protein, it would look the same. So in other words, it doesn't lean on any other part of the protein for its structure. It just seems to be a little chunk that sits there by itself, and it would be easy to remove it, and it would basically look the same. So that's your guys' introduction to protein structure, and I don't see any point to go further with it. I think the way you learn protein structures is by actually looking at proteins. So for practice, in addition to the worksheets, I'm going to suggest that you look at some actual protein structures. Manipulate them on your computer screen in front of you and see what you can see what kind of features you can identify. The best way to look at protein structures is to pull up the protein data bank, or PDB, and its website is www.rcsb.org. pdb.org, I guess, was already taken. And if you look at lecture 4.5, which is going to be posted soon, that gives you a brief walkthrough of how to use the PDB. The PDB is an invaluable tool for learning protein structure. If you figure out how to use the software, it can be a little bit challenging. All right, that concludes our talk on protein structure. We'll pick this up next time.